My name is Robert Alfano. I'm a distinguished professor of science and engineering at City College of New York. It's located in Harlem, and we cater to disadvantaged students and also to educate them into the state of the art. I've been involved since 1972 at City College, growing a ultrafast laser group called the Institute of Ultrafast Spectroscopy and Lasers. I joined City College in New York in 1972 after 8 years at General Telephone and Electronics in Bayside, New York. At my laboratory at General Telephone, I was involved in optical properties of semiconductors for my first 4 years. And then after that, I was switched into the laser group that was just formed at General Telephone to do basic studies of lasers. The first project was to look at the optical photon lifetime. That means vibrations in materials. How long do these excitations last in a material, be it a semiconductor or a liquid? So we started out looking at shooting in this picosecond laser pulse in green into a piece of quartz and calcite, and we found out that the light coming out changed frequencies into the white light that spanned 400 nanometers, which is blue light, to 800 nanometers, which is deep red. And that led us to the discovery of this continuum, we called it at first, and then it became the supercontinuum because it went beyond those wavelengths. And this particular area is now being celebrated at the SPIE's Photonic West as its 45th year since the discovery of the supercontinuum. The number of papers created since the early 70s is exponentially growing at this time, and the number of citations are up in the 60,000 range, and there's a lot of activity in using the supercontinuum. The supercontinuum has led to 2 Nobel Prizes. It's the enabling technology that offered Hensch and Hall the opportunity to do this frequency comb that be able to make a very stable ultrafast clock. Before that, in 1999, Zoel from Caltech used the supercontinuum to do chemical dynamics. So you have this white light, which is really been termed now the ultimate ultrafast light, because it's got all the colors, it's very short, and it's very easy now to obtain because of optical fibers. Optical fibers have allowed people to generate this supercontinuum with small little oscillators, and also big lasers, to generate this white light, to do various experiments in chemistry, biology, and now entering the biomedicine area. The supercontinuum generated from an optical fiber is the fact that you have this long length, and because of the long length, you can generate the modulation that is required to generate this white light. What happens in the generation of the white light is that it modulates its own phase. The laser modulates its own phase, chops itself, and by chopping itself, it generates all these sidebands that give you this white light. Using the supercontinuum in optical fibers, Japanese scientists have done terabits of information. It also is used in chemical reactions. What it is you have like a clock, and the clock is essentially the pulse width. The pulse width is the second hand of the molecular clock, and the clock is done by moving a prism or a grating to get you a time sequence. And so you pump, and then you probe at different times, and you see which molecules are excited or not. And it can be done in photosynthesis. You can see the various processes occurring in photosynthesis by probing the different wavelengths. And by doing the various wavelengths, you find out what excited states are excited. Or in a microscope, if you send in all the wavelengths, you could look at the time dynamics under the microscope to see which wavelengths are occurring that are exciting and energy transfer occurring within the molecular world. Getting back to General Telephone, we're in the lab, and I was working with Shapiro, who was also a young scientist at the time. And when I took the picosecond pulse of green light and shined it through a piece of glass, I found that our film, or where we measured the spectra, was all exposed. And I went into the office and I said to Stanley, I don't understand this. Well, something wrong here. So he says, go get some black tape. We'll tape up everything. So we taped up the spectrometer, just letting through a little hole. And we continued to do this. And every time we shot the laser off, I had to develop a plate and go into the dark room. And that's a very place that you don't want to be in this chemical, except if you're a photographer. And in those days, we didn't have video systems like we have now. And the laser was shot off once every minute. So you had to shoot and wait for the next laser shot. So each time we developed a set of film, we found the film exposed totally. All the colors were there. Didn't make sense at all. So we thought at first it was a leak in the room. Second, we thought it was the spectrometer was crazy, and so we had Gerald Ash come down to investigate what was wrong with the spectrometer. And it turns out they came back, they had perfect spectrometer, nothing wrong with the spectrometer. And then Lempicki, who was my boss at the time, he said, why don't you put up a Polaroid film camera? Strap it on to the, and don't go into the dark room. And let's have colored film. Let's see if it's real. So, and it turned out it was real. So we thought only at that time, it was only used for spectroscopy. And then over the years, when it became easier to generate with these fibers, people start talking about using this for communications, and FiOS came along, and people could use this for communications. And now, at this present time, it's going to be a bottleneck in computers. That the number of you need more bits of information. So more wavelengths give you more bits. More polarization gives you more bits. More whatever besides modes. Can give you more spatial modes, can give you more bits. So the idea is to create as many bits as you can, and I believe the supercontinuum, various portions, will be used in communications of the future. And if Verizon wakes up to know that it was discovered at their place, they don't even notice, because Verizon changed from General Telephone. And remember, each major company in the 1970s had major research going on. Today, that is not true. The research at companies are not as active they were in the 60s and 70s. There's a region that's not been touched, is using nonlinear optical effects on these biological materials. And really, the continuum has to go into the 300 nanometer range. Right now, these fibers and the crystals stop at 400 nanometers approximately. So 400 nanometers to about 2,000 nanometers is the continuum. And it's sort of, there's a stop at 400. But proteins, biological molecules, genes, DNA, which is really the aim, I think, of people in the photo business, for these photoscience, should really focus on the DNA, the genes, and that's in the 300 nanometer range, 250 nanometer range. So you have to generate this continuum into that range in order to do the spectroscopy of these building blocks of what's going on. I think neuroscience is the key. I think a big question left is, how do we think? How do we remember? How do we, I mean, it's unbelievable. Where's this stuff stored in our brain? And really, the only thing they've been using is NMR. And NMR is 1 millimeter resolution. The only thing is, light can go into the molecular world on a 1 micron scale, not a millimeter scale, not a 100 micron scale, but a 1 millimeter. So to probe the brain using light, I think, is the next horizon. There's a lot of room for doing. Light is a probe, and it could go in there, and you have all these colors, and all the colors could be done. If you excite the right dendrimer or axon or whatever you got in the neuron network, I mean, there's so many billions of neurons in your brain. It's much better than a computer, and it doesn't heat up. It's not, you don't have to worry about heat sinking it. I'm hot right now. But the point is, you don't have to, it's amazing you do all this analogs, not digital, analog computing in your head, and nobody knows how it's working. That's the next horizon. And I think that's going to be where you're going to focus the continuum, because it's the only source that has short pulse, all the wavelengths. Except you need, I think, you have to go down to the UV to do this. And the idea is multiphotons get you into the brain, multiphoton absorption. You do 800, you go for you cite 400, and you go into the brain deep, and I think that's the way to go.